Flowers can be beautiful, but their purpose is much more than giving us something nice to look at. At the University of Otago's Department of Biochemistry, Associate Professor Richard McKnight and his student Nathaniel Napier are finding out how genes and the proteins they encode for work.
My lab is interested in flowering time, how plants flower at the right time of the year. Legumes are really one of the, second most important agricultural family of plants. The legumes have the pulses, the beans, peas, the chickpeas, lentils. You grow a legume and that increases the amount of nitrogen in the soil. So Lucerne is one of those crops, either as a fodder crop just to feed your animals, or sometimes you just grow it and even just dig it in as to increase the nitrogen in the soil. Lucerne is related to the plant that we work on. It's another name is metacargo. This type of metacargo, this legume, is really an easy one to work with in the lab, and so there's international efforts to sequence its genome and provide resources. The key to the way we do our research is to identify mutants. We found in a population of metacargo plants one that looked a bit different from the others because it flowered a lot later than the others.
So here's a plant a bit earlier in development, and here's a plant later in development. And as you can see, the plant has produced some flowers.
Richard and Nathaniel decided to sequence the genes of the mutant metacargo plant and compare them with the genes of the normal plant to see where they are different.
It is very simple to get the sequence of all the messenger RNAs that might be in a plant.
To sequence the RNA, first of all you need to grind up leaves from the plant. Then you need to extract the RNA from the leaves.
Powderize the tissue within.
When the RNA is extracted, it's turned back into DNA so it can be sequenced. These sequencing machines convert the DNA into gigabytes of sequence data, ready to be analyzed. Richard and Nathaniel found a single mutation in a gene encoding a protein called Phytochrome Oberlin synthase. If you magnify a cell hundreds of thousands of times, you can see hundreds of thousands of proteins inside. But here are the two proteins that we are talking about. On the left is Phytochrome Oberlin synthase, and on the right, a photoreceptor. A photoreceptor is a type of protein that fungi and bacteria use to detect light. If you look inside the photoreceptor protein, you can see some small molecules called chromophores. Here they're colored pink. When you shine light on a photoreceptor, these molecules change shape, making the photoreceptor switch on a signal that tells cells to turn on flowering genes. The Phytochrome Oberlin synthase protein helps to make the chromophore molecules that end up inside the photoreceptor. But in the mutant plant, the gene for Phytochrome Oberlin synthase has a mutation in it that puts a stop codon in the middle of the gene.
Down here you can see that in the mutants it's changed to an A. So rather than a tryptophan, we have a stop codon.
That means the plant only makes half of a Phytochrome Oberlin synthase protein, and this protein can't make the chromophores needed by the photoreceptor to work. So no matter how much light shines on the photoreceptor, it won't turn on the flowering genes.
Even when it's in conditions of spring, then it thinks it's in the dark effectively, so that it flowers very late. So the way we set about proving it was to demonstrate that the mutation was linked to the phenotype, the late floweriness. So what you do is, it's easy in plants, you can get two plants, cross them together, pollen from one had it, so you get seeds, self those seeds to get the next generation, so it's segregating in the next generation. And then you'll have some plants that have the phenotype, late flowering, some plants that are just normal.
When Nathaniel checks to see if the plant has the mutation or not, he can't look at the DNA directly. He extracts DNA from the leaves and makes millions of copies of the gene using PCR. He then uses a special DNA cutting enzyme, which only cuts up the gene if the mutation is not present. To see if the DNA has been cut or not, he uses gel electrophoresis. First, he makes up a gel from agarose, which is a white powder made from red algae. It dissolves in near boiling water to make a gel when it cools. The gel sits in a tank of buffer. Nathaniel pipettes the DNA solution into a slot in the gel called a well. Then he runs an electric current through it. DNA is negatively charged, so it moves through the gel to the positive electrode. Little pieces of DNA move faster through the gel. Larger pieces get caught up, moving more slowly, so you can use the gel to separate out different sizes of DNA. The DNA has a special dye mixed with it, so you can see it in the gel using a UV light.
The size of the PCR is 350, and you can see in the mutant that there wasn't a cut. But in the normal plant, we can see that the product was cut, and we can see two bands, which is 250 and 100 base pairs.
So all of the descendants of the original mutant plant that were late flowering and had pale leaves also had the mutation that the researchers had pinned down.
So there's candidate genes that look really exciting, people haven't discovered before, that might come out of this work. So, fingers crossed.